All right. Hello, everyone, and welcome. So, you're about to head into the lab to analyze your very own DNA, right? Well, think of this as your ultimate guide. We're going to break down exactly how DNA fingerprinting works, step by step. That way, you'll know the science behind everything you're doing. Let's dive in and get you ready to make some discoveries.
So let's start with the big question, the one you're actually going to answer in your experiment. How is it possible that your DNA, which is 99.9% identical to every other human's, can create a profile that is totally and uniquely yours? I mean, that sounds kind of impossible, doesn't it? Well, here's the twist. The secret isn't in our genes, the parts of our DNA that actually code for proteins. No. Instead, it's found in these vast, non-coding stretches of our genome. For a long, long time, scientists just dismissed this stuff, even called it junk. But it turns out that so-called junk is where the treasure is hidden.
Okay, so to find that treasure, we have to look really closely at how our DNA is built, specifically at these repeating sequences that are scattered all over our genome. The specific kind of repeat we care about are called tandem repeats. All this means is that a short chunk of DNA bases gets repeated over and over and over again, right next to each other in this head-to-tail pattern. Just imagine a string of identical beads. The bead itself isn't what's special, it's how many of them you have strung together in a row. Now, these repeats come in a few different flavors. The ones you'll be looking at in your lab are called VNTRs. They're a type of mini satellites, and they have a pretty long repeating unit. Today, forensic scientists mostly use much shorter repeats called STRs. But here's the cool part. The basic idea you're going to learn with VNTRs, it's the exact same principle that all of modern forensics is built on. And that whole idea kicked off a revolution in forensic science. It totally changed the game. So let's take a quick look at how we even got here. So way back in the 60s, scientists started noticing these weird, super repetitive chunks of DNA. They didn't really know what they were, so they just called them satellite DNA. But the real breakthrough, the aha moment, came in 1984. A scientist, Sir Alec Jeffreys, realized that the number of these repeats was actually wildly different from person to person. He immediately figured out a way to see this variation, and he gave it the perfect name, DNA fingerprinting. This right here was the insight that changed everything. It meant that the unique code wasn't the DNA sequence itself, because that's often the same for everyone, but the number of times that sequence repeated at a specific spot, or locus, on a chromosome. That number, that became the key to our genetic identity. So let's get into the nitty-gritty. How exactly does just counting these repeats let us tell one person apart from another with such incredible accuracy? The entire technique really boils down to one key biological principle, polymorphism. It's a big word, but the concept is simple. It just means that for this one little spot in the genome, there are many different versions or forms out there in the human population. You might have 15 repeats, I might have 28, and your lab partner might have 40. It's this high level of variation that gives the technique all its power. Now, Jeffrey's original method was a lot of work. Seriously. It involved taking a person's entire genome and chopping it up into millions of pieces. Then you'd have to separate all those pieces by size and then use a special radioactive probe that would only stick to the fragments with the VNTRs, making them light up. The end result looked just like a barcode, and it was unique to every single person. The good news for you is that you don't have to do all that. Your experiment is much more elegant, much more focused. You're going to be zooming in on just one specific, very famous VNTR locus called D1S80. Okay, so for your lab, this is the magic number, 16. That repeating bead we talked about, at the D1S80 locus, it is exactly 16 base pairs long. Your entire job in the lab is to figure out how many of these 16 base pair beads you have on each of your 2 chromosomes. So, a few fun facts about D1S80. It's found on chromosome 1, and the number of repeats can be anything from 14 all the way up to 41. Because there's so many different possible versions or alleles, it's amazing for identification. And this exact spot on the genome, it was a total workhorse for forensic labs back in the early 90s. So you're basically doing a classic, historically important analysis. So how are you going to find your number? You get to use a way cooler, more modern technique. First, instead of chopping up all your DNA, you're going to use PCR to basically photocopy only the D1S80 region millions of times. It's like a genetic search and copy function.
Then you'll run those copies through a gel using electrophoresis. And this part is kind of like a race, right? The smaller DNA pieces, the ones with fewer repeats, they zip through the gel really fast. But the bigger, longer ones, they have more repeats, so they get tangled up and move a lot slower. The bands you see at the end, that's it. That's your unique number of repeats, your alleles.
And this PCR method was a total game changer for forensics. Why? Well, for one, you only need a teeny tiny amount of starting DNA, like from a single hair. It can even get a result from DNA that's been broken down over time. It is so much faster and more powerful than the old way of doing things.
So let's wrap this up by putting what you're about to do in the lab into the bigger picture of modern forensic science. Where does it all fit? Think about it this way. The D1SID analysis you're about to do is the perfect example of that foundational VNTR technology. It's what proved this whole concept could work. Modern systems, like the FBI's CODIS database, have just taken that idea and refined it. They use short tandem repeats, or STRs, which are even smaller. Because they're so short, they're even better at getting results from really old or damaged evidence. But the logic, the core idea, it is exactly the same. It's all about counting those variable repeats. So there you have it. You've got the history, you've got the science, you know the journey from so-called junk DNA all the way to the PCR technique you're about to use. Really, the only question left is the one you're going to answer yourself. What will your DNA fingerprint look like? Time to get in there and find out. Good luck in the lab.